Hello, I'm Christos Davatzikos. I'm with the Department of Radiology at the University of Pennsylvania, as well as with a couple of engineering departments, and I direct the Center for Biomedical Image Computing and Analytics, as well as the ABLE lab. I would like to share some of my thoughts on brain aids and some of the strengths, but also caveats of these types of models. So brain aids is, as we all know, essentially tries to fit a model to the data and try to identify people in red whose brain age is above their chronological age, and presumably these people have been developing disease or have advanced brain aging, and people in green who might have resilient brain aging or might have protective factors. And of course, brain age has been used as a biomarker in a variety of studies of schizophrenia, Alzheimer's, depression, sleep disorders, MCI, social anxiety, a number of others, where brain age has been shown to be an interesting biomarker that can be higher in most of these diseases. In brain development, it can be either lower or higher, depending on how you view brain age with respect to development. So that has kind of set up a race for challenges and comparisons of different methods, and that based on the mean absolute error, I boast that my method is the best of all because it achieves in 1.4 mean absolute error as opposed to 1.7, which is someone else's method. And we all try, or many people try, to beat these other methods by achieving even better and maybe even lower, even tighter fits using fancier deep learning methods with a lot of degrees of freedom and so forth. The point is that if we're interested in brain age gap as a biomarker of pathologies, it's not clear that tightly fitting brain age model tends to produce disease-specific markers. I apologize for restating the obvious or the well-known fact, but sometimes we tend to forget about these things. So let's say that we have a brain age model that finds, the gray areas here would be good features for predicting brain age in someone. However, these people have asymptomatic pathologies. And this cohort has asymptomatic pathologies, which can be anything from, small vessel ischemic disease, which will make the model exclude, for example, this area that might be related to this pathology. Then maybe they have TDP43, and then that might exclude another part of the brain that could be a good predictor of brain age. And maybe they have tau pathology that, again, might be asymptomatic or invisible in MRI, but it's there and might be changing brain age, or so might be actually making our brain age estimates worse than they would be. So we just exclude that as well. The model tends to exclude this. So it ends up being, focusing on areas that might not be affected by any of these pathologies. And of course, if there are multiple other pathologies in a standard brain, typical brain aging population, they will cause the model to try to avoid as much as possible and focus on things that might not be relevant to any of these diseases, actually, these pathologies. So a common argument, of course, that you hear is that we're supposed to use very, very super normal cohorts for building brain age models and then apply them to populations with pathologies in order to be able to better detect outliers. However, it's not clear to me that this is the way to go because, number one, it's very hard, or impossible, I think, practically, to find very, very normal cohorts, especially in studies of aging or adolescence. In aging, there's a bunch of pathologies that develop in our brains. Most of us have something in our brains, whether that's of vascular nature or Lewy bodies, tau, amyloid, things are accumulating, whatever. It could be causing, traumatic brain injury or mild traumatic brain injury, hypertension, diabetes. A bunch of pathologies have effect on the brain. It's very, very hard to find super, super normal cohorts, especially in large numbers. And if you were to find these cohorts, how relevant would be these models that we would derive from brain age to actually being able to detect pathologies? So we actually played with some of these ideas in the paper we had in Brain with Vishnu Basham, one of my bioengineering graduate students, and a number of other people, collaborators. So Vishnu at the time used one of the kind of high-end or state-of-the-art deep learning architectures, which was an adaptation of the inception resonant. And he basically used data from over 10,000 individuals from 13 different studies, as shown in different colors here, and he obtained very good cross-validated brain age gaps. I mean, this is not, he didn't make a particular effort to do anything fancy. He didn't harmonize the data. He didn't scope the data. He didn't pre-process the data. He just used two-dimensional sections just for the sake of having a generic model that generalizes well. So you get decent results, and then the results, the model generalized well to a new population in SHIP from Germany, from 3,000 people plus in Germany. So we were pretty happy about this. But then when you applied this model to people with Alzheimer's disease, with MCI, with depression, with schizophrenia, what we found out is what we actually expected, that the very tight fit, if you look at the effect size of brain age gap between patients and controls, you find that the effect sizes were larger, stronger for the not so tightly fitting models, which are the ones in the middle. These are the loosely fitting models, which in this particular case gave an effect size of, for example, 1.19. Then a moderately fitting model gave an effect size of 126, better, and then back down to 116 if you even more tightly fit brain age. And it goes down if you become even fancier, trying to fit the models even more strictly. Same thing was observed in MCI, in depression, in schizophrenia. As you see here, 0.63 goes up to 0.79, it starts to go down 0.76 as we start to fit brain age models more and more tightly to the data. So the bottom line is that if we're interested in estimating someone's brain age, the best model is the passport or the birth certificate. If we're interested in brain age gap as a disease biomarkers, then we have to evaluate it in the context of the disease of interest or pathology of interest, not in the context of mean absolute error. I think I really urge people, particularly those who organize challenges and comparisons, to do that. So I will keep talking about some caveats of brain age models just to make sure that we all have them in mind. I'm restating the obvious, but sometimes we tend to forget these things. So different pathologies have different effects on brain age gap. Say again that the gray are the regions of the brain that the brain age model finds as very good predictors for brain age. However, there's a pathology A that affects this part of the brain, which squarely sits in the middle of the gray area. So that is going to trigger the brain age gap. It's going to basically make these people look older than they actually are in terms of the brain age. But this pathology B barely overlaps with the model. So it's not going to trigger the brain age model. Is pathology B less severe than pathology A? Not necessarily. It'd be even more severe. It just happens to not coincide with the areas that the model found as informative for brain age calculation. So again, that's a caveat that has to do with sensitivity versus specificity. If the brain age is triggered by a pathology, then sure, there's something is happening there. But if the brain age gap is not triggered by a pathology, that doesn't necessarily mean that the pathology doesn't have an effect on the brain. So again, we have to have this in mind. And when we have negative results and brain age gap doesn't show any difference, it doesn't mean that nothing significant is happening in the brain. The one thing that was kind of a positive finding in our brain paper was that if you brain age models are good for pre-training and transfer learning. So for those of you who are familiar with deep learning, of course, we know that you have to pre-train your network, and typically people use ImageNet or some other large database, which is shown here in green. And then you go to train it in a smaller data set of Alzheimer's patients or MCI or schizophrenia patients, as we did here. And you have much smaller sample sizes, but because you have pre-trained and you transfer the weights of the network, you do much better. What we found is that if you use the typical pre-training using ImageNet, the green, it starts to do poorly if you then have a small number of patients to train on, like 100 people, for example, here, or 200 people. And these are not atypical cohort sample sizes in cohorts with patients and controls. However, if you pre-train on brain age and you apply it to schizophrenia, Alzheimer's, and so forth, you do better, consistently better, especially in smaller sample sizes for the most part. So we're also using this in the context of auxiliary learning, and we have found that essentially training models to estimate age does a good job in terms of finding a good starting point for further optimization for disease-specific networks. The final point I would like to make is heterogeneity. these two red dots that I've indicated here, these people deviate, they have the same brain age gap. They deviate from typical brain age in the same way, but by the same degree according to brain age gap. But they might have very different patterns of deviation from typical brain aging. And I'm illustrating that with some of the work that is going on in my lab by Divya Srinivasan and Matilda Antoniadis, who are doing a simple thing. They're just taking a two-dimensional system here where functional brain age is on the x-axis and structural brain age is on the y-axis. And of course, people who are at the top here will have a high brain age gap, whether they're orange or red. People who are on the right will have high functional brain age gap, whether they're red or green. However, the differences between these two, for example, take the red and the green. The red start with consistently higher amyloid level here, and they increase. The green start with significantly lower amyloid levels at the younger ages, and they build up as they get older. The green again have significantly lower rates of cognitive change over time compared to the red, even though they both have similar functional they have similar functional brain age gaps. And so forth. If we take, for example, people who deviate in terms of the structural brain age, which is the orange and the red, regardless of the functional brain age, we see that the orange and the red have very different characteristics. For example, if you look at the red here and the orange here, they have very, very significantly different future cognitive decline. This is from longitudinal data. Both of these groups would appear to trigger a brain age gap in a typical structural brain age model, but they are very different because their functional brain age is different. Same thing, if you look at here, they start from similar levels of white matter lesions, but they increase at very different rates, and so forth. So it's very important to understand heterogeneity and to understand that a single brain age index or even multiple of them don't really capture what is going underneath. And towards this end, we actually, last year, we published a paper, one of my students here, Zizang Yang, from the applied math program, developed this method we call SMILE-GAN, which I don't have time to go through the details. You can go through the paper, but it's a deep learning method that allows us to dissect heterogeneity between, in terms of the differences between a comparison group, say healthy controls, and a target group, say disease, or for example, resilient brain agers and advanced brain agers. So he did that in the context of MCI and Alzheimer's disease, and he found that there are multiple patterns of brain atrophy, despite the fact that this population was MCI, and that depending on which pattern you express, if you want, which pattern of brain atrophy you have very different future trajectories in terms of progression. Now, this was in the context of MCI. We are applying these types of methods in terms of brain aging and trying to kind of dissect the heterogeneity of brain aging trajectories instead of speaking about a single brain age gap. And we're talking about multiple dimensions along which people might deviate from typical brain aging. And then Ioana Scambardoni here has a poster in this conference, and I urge you to take a look at it because she has some interesting results. Finally, the last thing we really need to have in mind is size and diversity of training data. Spent too many years in the, between 2000 and 2010, 12, working with a relatively modest data sample size. And at that point, I realized that we really have to obtain very large data sets. So I put a significant amount of effort leading a number of consortia. This is the ISTG consortium where we brought together about 65,000 data from 65,000 people, 84,000 scans and longitudinal scans to understand brain aging trajectories. This is another consortium that was funded by NIH a couple of years ago that many of us co-lead. There are two consortia, actually, the AI for AD and the Phenotypic Harmonization Consortium. I'm missing a lot of faces here, but many of us put our efforts together to bring together over 50,000 MRI scans, cognitive phenotypes, and whole genome sequencing data, try to obtain better and deeper training of this machine learning model. So just to conclude, We have to evaluate brain age models in the context of the task at hand, whether it's diagnosis, prognosis, response to treatment, not in terms of the mean absolute error. Brain age models can serve as good pre-training or auxiliary tasks that prepare the ground, if you want, for disease-specific training. And a single measure doesn't save all. Basically, there's an ID, and that requires a very rich representation of data. So with this end, a kind of view of a lot of our work in machine learning as that of trying to build different dimensions of a brain chart, of a coordinate system that tries to find dimensions that are interesting in some way or another, whether these are disease-specific dimensions or deviations from typical brain aging or structural, functional productivity patterns or genomic patterns and so forth. Hopefully, we will break down heterogeneous diseases into more neurobiologically uniform subcategories, which perhaps have different prognosis, diagnosis, or response to treatment. Thank you very much.